Bienvenidos al canal del grupo de estudio de hormigas neotropicales. El día de hoy presentaremos la charla The Potential of Studying Morphology Using MicroCT, a cargo de Adrian Richard del Okinawa Institute of Science and Technology de Japón. Síguenos para más contenido relacionado con hormigas y no olvides suscribirte. Adrian is an insect morphologist whose passion since a young age have been ants. He wants to understand how and why all the many forms of ants that we see today came to be. At first step, during his graduate at the Jena University, Adrian worked on head morphology, and now, for his postdoc, he's continuing with the feeding system. His main research techniques are micro-CT scanning and 3D reconstruction. But Adrian also has worked a bit on ant paleontology and taxonomy and on morphology of a few other insect groups.
Listo, Calisto. Bueno, como ya vieron, hoy tenemos a un gran morfólogo de hormigas, Adrian Richter, que nos va a hablar un poquitico acerca de cómo implementar estas nuevas tecnologías como microCT, microCT, reconstrucción de microCT para entender la evolución, la morfología de las hormigas. Como ya vieron, él hizo el pregrado, la maestría y el doctorado en la Universidad de Jena en Alemania y actualmente está laborando como postdoc en el Okinawa Institute of Science and Technology en Japón. Entonces, para no quitarle más tiempo a Adrian, le daré la palabra. All right, Adrian, take it away.
Fantastic. Yeah. Oh, I can't share my screen right now. That is apparently deactivated.
Okay, try again. Sorry, that's on me.
All right. Yeah, no, I can. Fantastic.
All right, perfect.
Yeah, thank you so much for the introduction and for that super cool little video. That is really, I was very happy to see that. Thank you for that. Yeah, thank you for the invitation. I'm very happy to have this chance to introduce a little bit of my morphological research today. I apologize if my voice sounds a bit weird today. I have a bit of a throat ache. There's apparently like a weird bacterium going around Japan right now. So yeah, if there's any problem with my voice, I apologize. That's because of that. Yeah, so I will talk a bit about the morphological evolution of ants and how I research that using micro-CT scanning. Yesterday, going through my talk again, I realized that I will not actually talk that much about like the technique itself, micro-CT scanning. So if any of you have more questions about that, either ask me later or just stop me in the talk if there's anything unclear or you want to like hear more about anything. Yeah, right now I'm doing my postdoc at OIST in Okinawa in Japan, which is this nice looking institute in the middle of the forest of the subtropical island Okinawa. So it's a pretty nice place to study, especially because there's also lots of nice ants around in the forest here. So great for an ant scientist like me. And we also have nice equipment, of course, to do this kind of morphological work, to do micro-CT scanning and all that kind of stuff. Yeah. Now to kind of introduce the project that I'm going to talk about today, most of my projects that I've worked on so far are in some way insect morphology. So studying kind of the body shapes of insects. And the 4 projects that I will focus a little bit on today are, first of all, the evolution of ant head structures. That was my PhD project, as that the cute video nicely introduced already. Then I will talk a little bit about the anatomy of the digestive tract in ants. That is something that played a little role in my PhD project, but that I have kind of as my main project in my postdoc right now. But I actually don't have a lot of results yet. So I will only be kind of like talking a little bit about that, about kind of the direction that I hope to take that project in. Then I will talk a tiny bit about how I contribute to some taxonomic projects with micro-CT scanning. And then finally, I will talk a little bit more about a project that I'm working on right now. I actually have to finish the paper for that project, which is a bit annoying, but yeah, it's as good as done. So yeah, that will be kind of the up-to-date update on the mouse part interlocking or the mouse part closure mechanisms of ants. Yeah, and that's kind of the roadmap for today. Now to start us off, why am I even focusing on ants? And probably most of you, I don't have to convince that ants are actually pretty cool. They're pretty cool insects. But I still like to show this slide to demonstrate this a tiny bit. So all of the words that you see on this slice are just different words to refer to ants in different German languages and German dialects. And you basically see that everyone, all of these different like languages and cultures, all of them have a word for ants. And you could most certainly do this for languages all around the world. Except for maybe like Arctic circles or something. What this is just supposed to demonstrate is that all humans basically have a concept of what an ant is. We all recognize them and recognize them as kind of important, reflected in that we have a term for them. And this important is, of course, like because of their pure ecological dominance, they are super numerous and super important in basically all terrestrial ecosystems. And this kind of dominance and their social lifestyle has always had a lot of impact on humans and always fascinated researchers. And so that fascinated me as well, of course. But beyond that, beyond kind of the like ecological importance of ants, I also just find them pretty cool. Like if you look at this little like shiny knight or robot looking thing at the bottom, I don't know, I just think it has a cool body shape. I think it's a cool thing to look at. And that is probably one of the reasons why I am studying ant morphology, because I want to understand why their body looks the way it does and how these cool bodies of ants evolved. And that is definitely also kind of an interesting evolutionary question, because body shapes in ants, ant morphology is actually pretty damn diverse. We have, of course, roughly 15,000 known ant species and probably some more unknown ones. And among these, there's a huge variety of different shapes and forms and sizes and differently shaped weird structures that you can see just if you kind of look at this overview of a few different ant heads, just kind of looking at their faces. And the evolution of this huge diversity of these many different morphologies, how and why that evolved, that is kind of what drives my research and what I ultimately hope to contribute to finding out more about. And as a kind of morphologist or evolutionary biologist, an advantage that I have nowadays over a previous generation is that we now have a pretty good understanding of the phylogeny of ants. And phylogeny is, of course, kind of the basis to do any kind of evolutionary analyses to understand how features evolved. We first need to know how the organisms in questions are related to each other. Who is like closer related to whom. And for ants, molecular phylogenetic work has made a lot of progress in this area recently. And so we now have actually a pretty good idea how at least like the subfamilies of ants are related to each other. Although there's still no species or even genus level tree of all ants. That would be nice to have eventually. But in the meantime, there has been a lot of progress already that we can use. And kind of this progress has actually also like actually induced or like created some questions for evolutionary biology of ants, because these subfamilies of ants, this kind of lower level of the phylogeny, these individual groups, we actually have a pretty good understanding of these groups. So even when we just had morphological data, most of these groups were already recognized as kind of natural evolutionary groups. And so we have morphological features that are characteristic for these groups, that evolved in these groups. and that we kind of can recognize these groups from, even from just morphological data alone. But for many of these deeper nodes of the ant phylogeny, for many of these deeper splits in the phylogeny, we actually don't know that so well. A lot of these splits were only really recovered with molecular data and not really recovered before that with just morphological data alone. And to this day, for many of these nodes, for many of these splits in ant phylogeny, we don't have any morphological characteristics that really define these clades and that we know changed at these steps in ant evolution. And so that's, of course, one of my goals, to kind of better understand like the sequence of evolutionary steps across the ants. And the kind of main methods I use for that are, of course, to a large degree, micro-CT scanning to study the morphology of the ants. And so micro-CT scanning, basically, you have, just like in human medical micro-CT scanning, you have an X-ray source, and that X-ray source shoots X-rays through a specimen, through an ant specimen, and by taking kind of X-ray images of the ant in 360 degrees all around the body, we can get these nice reconstructions where we can kind of see the internal structures of ants. Can you see my mouse cursor?
Yes.
Or do I need to use it? Okay, perfect. And then once we have these kind of X-ray images, we can do a process called segmentation, where we kind of mark the individual anatomical structures in the head, for example, but it could also be any other body part. And once we have this segmentation, this allows us to get nice 3D reconstructions, 3D renderings, where we can actually visualize all of these internal or external anatomical structures and compare them in 3 dimensions. So we can really see the shapes of these structures. We can see where they are in the head, where they are relative to other structures, and we can compare these structures across different species. And of course, these kinds of 3D models also allow us to extract some extra information. We can quantify the shape of structures using 3D geometric morphometrics. We can just extract volumes of different structures. We can count muscle fibers or gland cells. We can determine muscle attachment angles. There's a lot of information that we can extract from these kinds of 3D data about our organism. But of course, I'm also using some more kind of traditional classical morphological methods, such as scanning electron microscopy to document surface structures of different different traits. And I also use histology, where you get histological sections through the ant body, which, like micro-CT scanning, also gets me the internal features, but the resolution that we can achieve with it is still comparatively limited. So some of the fine tissue details, like details of gland morphology or something, that is still easier to see in these kinds of histological slices. And then basically I use all of that morphological information to somehow code that. In the easiest case, I would just code a character matrix and to determine like features that are present or absent, for example, in different ant species, or rough shapes that are different in different species. But it could, as mentioned before, also be all kinds of other morphological data that is more quantitative. And then I can use that to analyze these characters in some ways. The typical analysis for evolutionary inferences would, of course, be ancestor state reconstruction, but there's also a bunch of other kinds of statistical analyses that you can do to understand better how and why your characters evolved in a certain way. Now, to get to my PhD project on the ant head, why did I choose the head as kind of my study focus? The head as a body part is, of course, rather important for ants because it has some very crucial character systems. First of all, it has some of the very important sensory organs, such as the eyes and the antennae for like mechanical sensation, but also chemical sensation, basically feeling and smelling around. And the head also, of course, has the mouth parts, which are the ant's main tool to interact with its environment, and of course also for food uptake. So one of the most important kind of basis of the ant life. And the head also has some classically recognized autapomorphies, so features that characterize ants relative to their ancestors or their relatives that kind of evolved newly in ants. And kind of the most classical features that everyone will think about at the ant head as like autapomorphies of ants are the geniculate antennae. So the ants have a relatively long first segment of the antenna, the scapus, and then they have a strong bend towards the rest of the antenna, the flagellum, which is relatively short. And the second feature is that the ant head is overall prognathous. And so this means that the ant is directed in the long axis of the body. They basically face forward with their mouth parts rather than downward or backwards than many other insects. And one of my goals in my PhD research was to explore these character systems a bit more and maybe add some additional apomorphies of the ant head to those that we already knew. And to do that, I studied ant heads across the phylogeny. Sorry. Kind of the main portion of the study was very detailed reconstructions of the full head anatomy of these 6 species of ants, which kind of cover all of the major clades across the Formicidae. And here I did really like detailed 3D reconstructions where I extracted the whole anatomy basically of the head. And you can see one very interesting specimen down here to the right, Gerontoformica. So this is a fossil ant species from the stem group of ants. This came from a 100 million year old Cretaceous age amber fossil. And these stem group ants are already ants, but they are not closely related to any of the ants living today. So they are potentially more close in morphology to the ancestor of all ants. And so this was a very kind of important data point for my PhD thesis because I got kind of a glimpse into the earliest evolution of ants. And the very cool thing about this specimen was that it had actually a lot of this internal anatomy, these soft structures, the muscles, the brain, the digestive tract, some of the glands. All of that was actually pretty nicely preserved in this specimen, despite its age. Like it's really like pretty remarkable, like 100 million years old, and we can still look at these internal features. And so yeah, that was one important data point for kind of my evolutionary inferences. And well, the taxon sampling here is mostly kind of spanning the deeper nodes of the ant phylogeny, so that I would hopefully be able to reconstruct some of the character transitions at these deepest nodes, like the very early evolution of ants. But towards the end of my PhD thesis, and some of this data is actually not published yet, I also looked at a lot of additional ant species across most actually of the living subfamilies, and also some outgroup taxa. And in these species, I didn't do these kinds of full anatomical reconstruction where I segment out and reconstruct every morphological feature, because that usually takes a lot of time. But in these species, I instead only looked at specific features that I identified kind of as potentially relevant in the more detailed reconstructions before. And so that kind of gave me some additional ability to say a little bit more about some of the deeper nodes of ants, and that kind of gave me a little more confidence of some of these character transitions. And in a way, this study was definitely successful. I did actually discover a lot of new potential like character transitions, odd apomorphies for these deeper nodes of ants. So this is a very simplified phylogeny. The names here, you will see like maybe some of you will know the study by Brendan Boudinot, where he kind of revised all of ants kind of systematics and the whole classification with the names of the different clades of ants. That is still not published. That's still on bioRxiv, but back then it just came out, so I used these names. But the Doroloformisia are basically just the poneroid clade that the formicoid clade that many of you probably know, and then the Poneria are the poneroid clade. And what I marked here on this simplified phylogeny in red are kind of new autapomorphies, so meaning autapomorphies of characters that had not really been studied phylogenetically before I studied them. So no one really knew that these characters could be potential autapomorphies. And purple are the ones that someone already studied before, and someone already maybe proposed previously that they could be phylogenetically relevant. And so you see that there's a lot of new features here that I discovered. And a lot of these new features are kind of fine anatomical details. A lot of them are internal features, anatomical features that are simply much easier to discover and to study if you have these kinds of 3D data, if you have like anatomical data and don't have to dissect and kind of photograph all of these species individually. I have to also mention that after I did this study, I had the chance at this point to look at some additional ant species, and some of these features that are kind of like still listed as potential autapomorphies here, I already know that they are not actually, or potentially they are not actually autapomorphies. So for this formicoid clade, for example, this is really the clade where it's very like puzzling for ant evolutionary biologists, like what is going on with this clade, because it's always recovered in molecular studies, but it was never before recovered in like other evolutionary studies. And so we really want to have some like autapomorphies for this clade. And I thought I found some, but now I looked at some additional ant species and it's probably more complicated again. Like these features that I found are probably more variable across the ants than I initially thought. And so, yeah, I will still have to refine this analysis and we'll have to do more tests to really find out if there actually is something that I can confidently say as an autapomorphy here. But yeah, I don't have time to go into all of these characters in detail, but I want to focus on one a little bit more because I think it's pretty interesting and is actually this kind of inspired my current interest in the digestive system of ants. And this is definitely also kind of like an ant autapomorphy. So I'm pretty confident about that. And that is the shape of the configuration of the pharyngeal sucking pump. And first, I want to look at this sucking pump in a typical wasp species. So this green kind of tube here, this green sac in the head, is used by insects to kind of suck food into their digestive system from their mouths. This is kind of the throat of insects, you could say. And the feature that I want to direct your attention to are these dark green bars at the side of this sucking pump. These are the so-called oral arms. And in wasp species, such as this Ampulex, which is kind of like a digger wasp, these are just straight bars, very simple structures. You see that there's this little wing at the end. And the function of these oral arms is basically to stabilize the sucking pump, but also to serve as point for muscle attachment. So there's this longitudinal muscle that connects from the oral arm to the front of the sucking pump. Then there's this transverse muscle that connects the oral arms on the two sides. And then there's a kind of a back muscle and a front muscle going to this posterior process of the oral arm. And now I want to look at that same structure in a typical ant species. So this is in a Brachyponera, so like a ponerine ant. And you immediately see that the configuration is a little bit different. So first of all, these posterior processes are kind of like enlarged, where these front and back muscles attach to, and they kind of project more sideways from the sucking pump. Then we see that the transverse muscle is attaching to this like dorsal plate, to this dorsal expansion of the oral arms. And we can especially see this if we go back to the side view. We see that rather than just being a straight arm, this arm kind of like curves upward and has this dorsal expansion, this dorsal plate. And this plate also gives additional kind of surface structure, additional attachment structure for this longitudinal muscle, which is pretty massive in ants usually. And so these are all kind of like autapomorphic features of ants that I can reconstruct. This dorsal expansion, the kind of modification of the posterior process, the enlargement of this longitudinal muscle, all of that is definitely features that ants generally share. However, there is also some variation within the ants of the sucking pump. And very interestingly, a lot of the formicines and also dolichodorines, such as this formica here, which are kind of ants that mostly feed on, that are more kind of herbivorous, that feed a lot on liquid plant kind of based food sources, such as extrafloral nectar or like aphid kind of products. They have a more simple construction again. So if you look at the oral arm here, it's more of this straight bar again. These dorsal expansions are much smaller, and the posterior processes are also not as like sidewards directed. Whereas in all of the other ants that are more kind of still predatory, we have these massive dorsal plates, although they are also differently shaped in different groups. Like they are not always exactly the same shape. But generally, we have more of this dorsal expansion. And so I think that these differences in the sucking pump anatomy in ants are most likely related to these differences in diet that we find in different ant groups. And I think that gaining this more additional complexity in ants in the first place is probably because they are ancestrally predaceous, and where like most other hymenopterans, most wasps, for example, will well, a lot of them are predators, but they feed the prey that they catch mostly to their larvae, to their young, whereas the adults feed a lot on nectar. And ants, at least ancestrally, feed basically fully on just other insects. Both the adults and the larvae don't really take any other food. So that could be one reason why ancestrally ants first got this kind of more complex shape, if it really does have something to do with predation. I still have to test that more. But then some ants that got more nectar feeding again, those kind of reversed that. And well, this sucking pump variation is only one aspect of the digestive system that is pretty interesting in ants. There's also other interesting variation going on, for example, in the glands in the head, in the musculature of the sucking pump that I didn't go too much into today, yet. And we also have other interesting features such as the proventriculus, for example, which kind of connects the social stomach of the ants and the actual digestive part. And kind of analyzing the evolution of this whole system is the goal of my current project, where I want to utilize the AntScan data set, which has been generated here in the lab of Evany Konomo in Okinawa, where they scanned basically around 2,000 different ant specimens, hundreds of different species. And so we have a huge amount of morphological data now. And I want to extract that morphological data from these data sets of the digestive system to get a detailed reconstruction of the evolution of the system, but also to test hypotheses of correlation and hypotheses of kind of correlated evolution between the digestive system and the different lifestyles and ecologies of different ants. And yeah, I haven't gotten very far in that project yet, so I can't tell you much. This is kind of what it looks right now. Like I have a few of these sucking pumps, a few extra sucking pumps segmented out. These are just like very quick and dirty renderings. And you definitely see that they are like relatively variable. For example, in Campanodus, and this is a general feature of formicines, most formicines, they have this very like large back muscle at the sucking pump. And I think this could again like be a feature that could be related to their very liquid feeding diet and kind of social behavior, where they do a lot of prophylaxis, sharing liquid between different ants. But it's a bit tricky because on the right here is two myrmecine species that are also pretty liquid feeding, and they don't necessarily have this muscle as enlarged as the formicines. So yeah, maybe they have other parts of the system that are adapted to liquid feeding. The cephalotes here at the top is like super weird, like that is very unusual. You see that there is this huge like kind of ball, this huge kind of balloon-like expansion of the sucking pump behind these muscles. And yeah, I found that in several cephalotes species, but not really in any other ants so far. It fills out a pretty big portion of the head, actually, this balloon-like expansion. And cephalotes are generally a bit weird, like as ants. A lot of them like to feed on bird poop, and they also have a weird proventriculus shape, which is probably also related to that weird diet somehow. So yeah, probably something weird is going on with feeding in these ants, but so far I don't really know what this balloon could be for. But yeah, in general, I think this sucking pump, like the digestive system, is pretty interesting in ants. And I hope in the future I can tell you a bit more about it. Yeah, then short intermission, just I want to quickly mention this because I like this project. I was at the ant course in Vietnam last year, no, 2 years ago now. And I found two new species of leptanillines, a leptanilla and a protanilla. And together with colleagues from Vietnam and the United States and also Japan, we are describing these two new species and additional some other new species of the leptanillines from Vietnam. And as part of that study, we are also providing 3D reconstructions of the holotypes, where you can really look at the holotype in 3D in all of its kind of glory. And these 3D reconstructions definitely helped us to kind of look at some detailed features to compare these species and to, yeah, kind of identify the species. But now, finally, I want to get a little bit into my current ant mouse part projects. And if we talk about ant mouse parts, then the kind of most prominent features that everyone sees and everyone is aware of and a lot of people study is kind of the mandibles here at the tip of the head, as kind of like the main grabbing and cutting and grinding tools of ants. They are obviously super important and they are very diverse. But below the mandibles, there's also a bunch of other mouse parts that are also important, I think, and that I'm focusing on right now. And these are kind of the maxillolabial complex. So this bunch of complex structures here is called the maxillolabial complex. The dark brown structure in the middle is the labium, which is kind of the tongue of ants used to lick up food. And then at the sides, these brighter structures are the maxillae, which have these shovel-like structures, which are used to manipulate food, kind of shovel it in towards the mouse. Also they actually help with cleaning, so with self-cleaning or cleaning other ants. They also use these mouse parts. And then we also have these finger-like extensions, which are the palps, which are used for sensing. So like similar to the antennae, they are chemical and mechanical sensory organs. But most of the time, you actually can't see any of these complex structures because they are all hidden behind this blue structure, which is the labrum. And the labrum is just kind of a solid cuticular plate that sits in front of these other mouse parts. And in general, in most insects, it sits in front of the other mouse parts. But in ants, it especially sits very tightly in front of this maxillolabial complex, covering these complex and soft tongue and shovel parts and kind of protecting them. And this tight fit between the mouse parts is kind of enabled by a novel interlocking mechanism. So you see here that the labrum has this basal process, this basal hook, and the maxilla, when the mouse parts are closed, kind of presses against this hook actually. Like part of the maxilla is pressed against this hook, and that locks the whole labrum in position in front of the other mouse parts. And that kind of pulls the labrum really tightly on top of the other mouse parts. And so the mouse is really tightly sealed off in ants, which is not a feature that you have in any other insects that I'm aware of. Yeah, here's just another image of that. Like I just took that directly out of the paper that I'm writing right now. In the open condition, like everything is kind of loose, but then you close your mouse parts, this kind of projection of the maxilla presses against the labral hook and then keeps the whole labrum in position. And all of these soft, kind of flexible, potentially vulnerable structures, like all of this is now hidden behind the labrum. And why could that be important for ants? Well, I think it just helps them to protect these potentially vulnerable mouse parts, because ants do all kinds of stuff with their mandibles, some pretty like potentially dangerous stuff like fighting prey insects, digging in the ground, fighting other ants, just a lot of different tasks. And through all of these different tasks, potentially these softer, flexible parts of the maxillolabial complex could be damaged, or they could just be like, there could be wear on them. And so, yeah, the ants with this mechanism can nicely keep them out of the way while they do other stuff. Now, interestingly, these mechanisms of mousepart closure are also actually kind of diverse across the ants, and they vary in the tightness of this interlocking. And the first one who discovered this and who had a lot of data for this was Roberto Keller, who is also, who I'm collaborating with in this project. And he initially imaged about 100 species using scanning electron microscopy, where he documented this mechanism. And now I added about additional 70 species where we could kind of see this mechanism a bit better in 3D based on micro-CT scans, and like documented some additional diversity of this mechanism across ants. And really, there's actually quite a lot of diversity of how exactly this interlocking like works. Generally speaking, there is two main things that we can observe. In some ants, like there is this projection of the maxilla, of the maxillary stipes, to be precise. So this is kind of the central hard cuticular plate of the maxilla. And that kind of projection of the stipes presses against the labral hook, which leads to a very tight interlocking. Everything is nice and locked. But many ant species also have actually no such projection of the stipes. And instead, the interlocking is much more loose and only interlocking between the pulpus of the maxilla. And so the pulpus, as I mentioned before, is kind of a movable finger-like structure. And so this is, of course, not as tight as if you have a solid structure pressing against the labral hook. And so, yeah, these different configurations lead to some different closing levels. And yeah, I won't go into all of this detail too much right now, but like, so suffice to say, some ants have more loosely interlocking mouse parts and some ants have very tightly locking mouse parts. And if we reconstruct the evolution of these differences, then we can see that the loose kind of condition is retained across most of the kind of backbone of the ant phylogeny. And then it was specific clades that switched towards this tighter interlocking. That is mainly the whole poneroid clade. It is the subfamily Dorylines, which includes the army ants. It's the subfamily Ectatomines, and then a few other individual clades like many myrmecines, Myrmecia in the myrmecines, and some formicines as well. And now the question is, of course, why some ant lineages, some ant groups went towards this more tight interlocking, but also why not all ants just went to tight interlocking, why some of them kept the looser condition. And so why more protection is needed, of course, my main hypothesis was that this had something to do with lifestyle, and specifically with lifestyles that would have something to do with ants' need to protect their mouse parts. And so the most obvious situation where this might be important is in diet and feeding. And so the clearest kind of hypothesis was that hunting ants, like predatory ants, probably need this kind of extra protection because they are combating like their prey insects, and prey insects are like made of hard cuticle. They are sometimes maybe defensible, have some defensive strategies. So while you fight them, you probably need to protect your mouse parts. But alternatively, it could also be about diet more generally and more about what they feed on. Do they feed on like liquid food sources, or do they feed on hard food sources? So do they have to process hard prey objects, or do they have to process hard seeds, for example, as plant-based food that is also pretty hard? Or do they only mostly consume liquid food sources? And so these were the kind of two codings that I used for diet. And then I used some proxies for ant aggressive behavior, which are kind of like imperfect proxies because there's not a very good data set about ant aggressive levels, like how aggressive ants are against other ants, for example. That is not as well documented as some of these other features. So I use these kind of as proxies for that. For single versus group foraging, where kind of my idea was that if you forage in a group, then maybe you're kind of protected by being in a group. Whereas if you're a single forager, you really need kind of personal protection. And that may be important. Stinging versus non-stinging. Yeah, if you're a stinging ant, then you have to go into kind of close quarters combat with your opponents, whereas non-stinging ants may have other strategies. And then large and small colony sizes. Large colonies probably don't have to care so much about each individual worker. If they lose some individual workers, it's fine. Whereas smaller colonies may need really better protection for each of their individual workers so that they don't just die. Yeah, so these were kind of the overall hypotheses that I wanted to test and that I had data on. But I do also have to mention that like the ecological data for ants is still, or even though there is quite few databases, at this point, the data is not great. So I had to do a lot of assumptions for some ant species. But be that as it may, some patterns are actually pretty clear. If we look at like the different like states of ecology of these features that I considered, and then look at how many of those that are, for example, a hunting ant have tight interlocking and how many have loose interlocking, and how many that are not hunters have tight and loose, we pretty clearly see like a correlation between hunting and being a tight interlocker. And if you're not such a hunting ant, then you don't need such tight interlocking. And similarly for liquid feeding, this also is pretty clear, like the correlation doesn't seem as strong. And then for foraging mode, it's not that well correlated. Stinging is also kind of correlated here, and colony size is, well, not really well correlated. And if I actually test these relationships, these correlations, with a method taking phylogeny relatedness of the species into account, I used phylogenetic logistic regression for this, then yeah, predation absolutely comes out as the most well-supported kind of predictor of mousepart state statistically. Like it's the most significantly supported. It's kind of some best fitting model based on AIC. It has the strongest kind of coefficient. So the effect of predation is the strongest. Like predatory ants have 20 times higher odds of being tight interlocking than non-predatory ants. So yeah, I think predation is at least one important kind of driver of this evolution towards tighter interlocking in ants. But then why don't all ants have just tight interlocking? Because yeah, okay, sure, the very predatory ones need it more, but all ants are kind of aggressive and kind of would benefit from protection, right? So why don't all of them have it? So here my idea was that maybe there's a trade-off between protection and some other mousepart functions. And I used pulpus lengths as a proxy for this, because I noticed that a lot of the tight interlocking ones had a very short pulpus actually, whereas the ones that had loose interlocking had a more longer pulpus. And the pulpus is an important sensory organ, of course. And so if you have it shortened, then you just have less of the sensory capabilities and can use it less. And yeah, here as well, the statistical association was pretty clear, also with taking phylogeny into account. Yes, pulpus length kind of predicts tightness level, and tightness level also kind of predicts pulpus length. And so, yeah, I think there could be just a trade-off of, if you have tight interlocking, you need to kind of reduce other mouthpart functions. And so I think these two drivers, like trade-offs on the other hand, and then need for protection on the other hand, yeah, those are probably two of the drivers of this mouthpart interlocking. Probably there's other features, other relevant evolutionary mechanisms, but those are the ones that I was able to detect. Yeah, coming to the end of my long-winded discussions, I hope that I was able to convince you that with these kind of morphological studies, we are still able to discover new character systems for evolutionary and systematic works, where we can still find out something about evolution of ants, for example. Then the 3D methods also help us to better understand the evolution and function of very specific character systems, such as the digestive tract, where I don't know a lot yet what the function actually is of the variation, but also of the locking system here as another example where I now have a pretty good idea of what the functional impacts may be. And in general, I think micro-CT scanning still has a lot of untapped potential. There's a lot more information that we can extract on these data sets. And nowadays, more and more of these data sets are becoming available, and they are also slowly becoming available to a wider range of scientists. And I think as they become more available and as more methods get developed and as more people use them in their research, we are going to be able to extract a lot more information and yeah, still do a lot more with these kinds of data in all kinds of evolutionary and systematic studies. Yeah, and with that, I want to thank all of you very much for your attention and for coming to this little talk today. And I want to thank everyone who made my research possible, who collaborated in my projects, and who gave me the money to do them, actually. So yeah, thank you very much. And I'm open for discussion and questions.
All right, thank you so much. Muchísimas gracias, Adrián. Qué charla tan interesante. Qué chévere todos esos modelos. Entonces, listo, damos apertura a la sesión de preguntas. Si alguno tiene una pregunta, pues está libre para levantar la mano y hacerla directamente. O si quiere enviarla por chat, yo se la relevo a Adrián. Okay, so we have a question from Maria Tocor Alonso, who says, "Great talk, Adrian. Amazing work. I'm wondering, how do you select the species you use for CT scanning? Also, if someone is interested in collaborating with you, how can they contact you? Thank you so much."
Yeah, thank you for the question, Maria. So the species to select always kind of depends on the research question, generally speaking. Can I go to, yeah, okay, I will just go back. So in my PhD thesis, part of my selection process was just what species do I have available, actually, because my museum where I worked is not like really focused on ants at all. So we didn't have a big ant collection. So it was more about what species can I kind of get my hands on, what species can I get. And then the second consideration was to span the phylogeny. So if my goal is to kind of say something about this node in the ant phylogeny, then I need to at least have some species of this clade and some species of this clade. Or if my goal is to say something about this node in the phylogeny, then I at least need some species from this clade and some species from this clade. And the more I want to say about the more nodes, the more species I need, of course, to cover this, like evolutionarily. But then, of course, there may also be specific functional questions. So with mouse part stuff, for example, I try to select species with different lifestyles. So I try to select species across the phylogeny, covering different clades of ants, but also species with different lifestyle, like choosing a myrmecine predator species, choosing a myrmecine herbivore species, and that for as many different clades as I could, right? And so the two main considerations are always like phylogeny, relatedness, and then functions. And then kind of the research question, what kind of function am I looking at and which species are kind of interesting from that kind of functional perspective. And then what species do I have like specimens or data available for? Yeah, for collaboration, I have an email address. I don't know, I don't have it on my slides, I think. But you can probably find it online. I have several email addresses. I have one with my with Okinawa Institute of Science and Technology right now, which is a.richter@oist.jp. Or like my standard email address that's also in most of my papers is adrichter@gmx.de. Very old email address, made that when I was still a high school student, I think. Yeah, or on Twitter. I'm also on Twitter. And yeah, and so yeah, collaboration. I always like to collaborate because well, not everyone has access to these kinds of micro-CT scan data, but if possible, I like to give people access if they are interested in it through like having kind of access to micro-CT scanning facilities. And also this ANSCAN project, for example, is going to be fully open access, so everyone will be able to access that. And if any one of you or anyone that you know of is interested in working on morphological evolution and wants to kind of learn how to do this kind of 3D workflow, I'm always happy to like yeah, help and try to try to like give some tips on how to do that kind of work. So yeah.
Awesome, Adrian. Just there's another question, but really quickly, would it be okay if I send your email through the Zoom chat?
Oh, absolutely. Yeah.
Okay.
Yeah.
All right. So I'll be doing that. And Camila as well asks, says, great. I was also wondering if there are any specific organs like the bacteriocytes that can be captured with CT scanning.
Yeah, so that's definitely something that I'm also interested in, these like bacterial pouches and stuff, right? So yes, generally speaking, that the pouches can be captured with micro-CT scanning. So we could see where they are, like how big they are, how they look, that kind of stuff. But micro-CT scanning is still kind of like limited in the resolution. So yeah, like the general location and shape is probably all we could extract in terms of information for these pouches. We can't like actually visualize the symbionts in there or something, like the actual bacteria in there. So yeah, the information that we can extract is limited, but we can most likely like at least find them in the micro-CT scans if the kind of if the specimen is well fixed and if the resolution of the micro-CT scan is good enough. Yeah, that's definitely something I'm also interested in exploring more in the future.
Great, thank you, Adrian. Andres Morales says excellent work, and we have a new message here from Julio, who says, hi, Adrian, great talk, thanks. 2 questions. Has Brownie Messia been studied through micro-CT? Is the technology efficient for non-amber ant fossils?
So as far as I know, Brownie Messia has not yet studied with micro-CT. That would definitely be super cool. I hope that happens at some point. Yeah, someone would have to get their hands on the specimen that has access to micro-CT scanning, basically. And yes, this can also be used for non-amber fossils, like for compression fossils or other kinds of fossil types. It is a bit more tricky to make that work. It depends a lot on kind of the stone or whatever the specimen is embedded in. But actually, just this Monday, we had a talk here at OIST from someone studying kind of super old arthropods, like early like even Cambrian age, like arthropod fossils, and they were able to extract like the legs of one of these stem arthropods from a compression fossil. Like you could only see the back of the fossil from the stone matrix, but they did micro-CT scanning on the whole fossil, and then they could actually reconstruct the legs that were previously like hidden within the stone matrix somewhere. And so yeah, this definitely works. but it is more difficult to get to work than amber fossils. Amber fossils are usually very straightforward. Other fossils are a bit more tricky, but it's possible, and I think there's a lot of potential there for still future studies.
Awesome, Adrian. I was also going to mention the case of, I think it was a couple of months ago, where I believe there was a paper where they did that with a spider fossil, and they were able to reconstruct it. Can't remember from where it was though. I think it was Chinese. There's a lot of this advances in micro CT technology with fossils is from China.
Yeah.
Yeah. Okay, so we have another question from Daniela. It says, excellent talk. From the digestive system, did you find any variations?
So yeah, I have, like, if you consider like the sucking pump, I've definitely already found a lot of variations, and I've mentioned some of those in the talk. Like the muscles in general of the sucking pump are different. The shape of these oral arms of the sclerotized hard parts of the pump are different. And then there is some like well-known kind of classical variation known for the proventriculus. Maybe I can go back to the proventriculus thingy. Oh yeah, that was too far. Yeah, well, the video will maybe play. Yeah. So the proventriculus, this part in between of the crop and of the midgut, is definitely variable across ants, and we can nicely see that usually in micro-CT scanning. But yeah, I haven't looked at that variation a lot yet. Like, I haven't really studied enough species. And I think there's probably also a variation in like the other parts, like just the size of the midgut, the size of the crop, the number of Malpighian tubules. All of these features are probably variable, but those are also a little bit difficult to quantify, because they are like soft structures, right? And they're elastic structures. So they can look very different in different ant species, in different ant specimens, not necessarily because it's like different in different species, just because it was like at the point when the ant died, it was like in a different shape, because it's elastic and flexible. So those parts are a bit more difficult to compare. And also, I just don't have enough data yet to really compare them. But yeah, in general, there's definitely variations of the digestive system that are interesting, and that I hope I can quantify more in the future.
Okay, awesome. I actually have a question too. Recently, I was speaking to a researcher at the AMNH who works with vertebrates, and was talking about how they had developed some tools to using like micro CT scans and stuff that are heavily deformed, reconstruct what the original shape of the organism was. And I asked like if they knew of any way of applying this like more broadly, and they said that it was very likely because the software originally was used more for mechanical things, for like literally like car decompression and stuff like that. And so I was just like wondering if you had also like dabbled or thought of applying these sorts of tools to fossils, because as we know, like fossil specimens a lot of times are completely desiccated, and so it makes it very difficult to study them.
Yeah, totally. I have definitely noticed that in the few fossils that I have worked with, there's always some level of deformation. And I mean, sometimes this even happens with like extant specimens. Someone deformed the gaster a bit or something. But yeah, I have not actually dabbled with that. Like, that's super cool that they found some better techniques to do that, or algorithms to do that with vertebrates. I have seen stuff like that before, like in invertebrates especially, that they have managed to kind of correct that a little bit. But usually it's because you have like two bones from both sides, right? And then some part of the bone looks good on one side of the animal and not good on the other side, and then other parts it's a reverse, and then you kind of use both sides to get kind of the overall ideal best that you can. So I know of like some of those methods, but yeah, no, I haven't really played with these kind of deformation or like correcting deformation methods myself. But yeah, definitely super cool and important for fossils, and it's nice if there's like new stuff being developed there.
I
should
think,
look into.
yeah, in the risk of turning this into a podcast, I think it would be a wonderful idea to use this ant scan now that we have it there on screen.
Mhm.
And in the age, sadly, we have to embrace it of artificial intelligence
Yeah,
to find a way to develop predictive algorithms, because it would be
absolutely.
absolutely amazing, not just for the amber fossils, but as well for, as we were mentioning before, for the impression fossils, to be
Yeah.
able to reconstruct what they were like in life.
Yeah. Yeah, like kind of I can definitely imagine some experiments where you take like live modern ants where we have the perfect shape, and then you deform those somehow, and then you train an algorithm to kind of recognize those deformations and to make it nice again. And then once those algorithms work nicely on the ants where we know the real shape, we can try to apply them to ants where we don't know the real shape, where they are just present as deformed specimens. And when maybe we can actually get like a much nicer idea of how they may have really looked like. Yeah, absolutely. I mean, AI for all of this, like data processing, like extracting information from CT, I think AI will play a very important role. I'm also not sure how I feel about that, but it will be useful for science, I think, at least. So yeah, it's probably a future direction that will be very important.
Yeah. Okay, well, let's give it a little bit, like maybe 1 or 2 more minutes to see if anyone else has any more questions.
Thank you for all the questions so far. I'm happy if my talk was somewhat interesting to people.
Okay. I think actually, yeah, we've had you here for more than an hour. And yeah, well, a lot of people might be wanting to head out. It's very late.
It's late for you guys.
Yeah.
Sorry for taking up all of
But
your
thank you.
evenings.
Thank you so much, Adrian. This was a wonderful talk. It's great getting to know the kind of technologies that are being used for like furthering ant studies. Everyone here is, well, not everyone, but most people here are ant people or ant adjacent people. So it's really cool seeing how these modern technologies are being applied, and hopefully this gives everyone in the audience, ideas on what sorts of things they can do, and they see that these things are possible, collaborations are possible, and even if you don't have a micro CT scanner because they are prohibitively expensive, through collaboration they might get to play around with this stuff. So thank you again,
Thank
and
you. I will stop this. And yeah, thank you all again very much.
vale. Entonces,
Good evening.
para todos, con eso cerramos. Entonces, de nuevo, muchísimas gracias para Adrián, fue una charla maravillosa. Y sí, hasta luego a todos.